In a splicing pathway, a number of steps may differ slightly in their order or might even reverse, but the pathway reveals the series of events undertaken by the spliceosome to drive the splicing reaction in the cell. Initially, the 5' splice site is recognized by the U1 SNRP. U2AF is made up of two subunits. The larger 65 binds to the PY tract and the smaller, which is 35, binds to the 3' splice site. The former subunit interacts with BBP SF1 and helps that protein bind to the branch site. This arrangement of protein and RNA is called the early E complex. So early E complex is formed by the joining by the attachment of all these proteins and SNRPs. So this is the pathway, and you can see here this is pre-mRNA, then one SNRP is attached, then other, and one is removed, another one is entered into it, then these two are present, and then this is a group, smaller group of proteins, and then this brings in, and by attachment of this group, a bulge is formed, this bulge, and this bulge causes the A to get closer to this 5' splice site, and then this is removed, and then finally this intron is removed. So these are the different steps in a splicing pathway. The former subunit interacts with BBP SF1 and helps that protein bind to the branch site. This arrangement of proteins and RNA is called the early E complex. U2 SNRP then binds to the branch site, added by U2AF and displacing BBP. This arrangement is called the A complex. So by binding these new SNRPs, the E complex is transformed into A complex. The base pairing between the U2 SNRP snRNA and the branch site is such that the branch site A residue is extruded from the resulting stretch of double helical RNA as a single nucleotide bulge. A bulge is formed, as you have seen in the figure. This A residue is thus unpaired and available to react with the 5' splice site. The next step is a rearrangement of the A complex to bring together all these 3 splice sites. This is achieved as follows. The U4 and U6 SNRPs along with the U5 SNRP join the complex. So these more RNAs and SNRPs, they join this complex. Together these 3 SNRPs are called the tris SNRP particle. Tri-SNRP particle, sorry. Within this particle, the U4 and U6 SNRPs are held together by complementary base pairing between their RNA components, and the U5 SNRP is more loosely associated through protein-protein interactions. With the entry of the tri-SNRP, the A complex is converted into the B complex. So you have seen in the figure a heavy complex, that is the tri-SNRP, that comes into the machinery, and then this complex is transformed into B complex. In the next step, U1 leaves the complex and U6 replaces it at the 5' splice site. So U1 is removed and U6 comes in. This requires that the base pairing between the U1 snRNA and the pre-mRNA be broken, allowing these U6 RNA to anneal with the same region. Those steps complete the assembly pathway. So all these steps complete the assembly pathway. So this is the sequence of events. So this is pre-mRNA, U1 attaches here, U1 attaches, then U2AF comes in, and then there is slight rearrangement, and then this E complex is formed, then this one is A complex, and finally when this tri-SN or SNRP comes, this becomes the B complex. So this is the transformation from early E complex to E complex, then to A complex, and finally to B complex, and then this B complex will rearrange to the C complex, and then catalysis will take place. The next rearrangement triggers catalysis and occurs as follow. U4 is released from the complex, allowing U6 to interact with U2 through RNA-RNA base pairing. This arrangement, called the C complex, produces the active site. That is, the rearrangement brings together within the spliceosome those components that together form the active site. So the different components, they are brought together in the spliceosome, and by bringing these together, the active site is formed. The same rearrangement also ensures that the substrate RNA is properly positioned to be acted upon. So this also brings about the proper positioning of the RNA. It is striking not only that the active site is primarily formed of RNA, but also that is only formed at this stage of spliceosome assembly. Presumably, this strategy lessens the chance of aberrant splicing. That is, it lessens the chances of wrong splicing. Linking the formation of the active site to the successful completion of earlier steps in spliceosome assembly makes it highly likely that the active site is available only at the legitimate splice sites. Formation of the active site juxtaposes the 5' splice site of the pre-mRNA and the branch site, facilitating the first transesterification reaction. So the positions they are arranged such that the splicing can take place, or first transesterification reaction can take place. The second reaction between 5' and 3' splice site is aided by the U5 SNRP, which helps to bring the two exons together. The final step involves release of the mRNA product and the SNRPs. The SNRPs are initially still bound to the laureate, but they get recycled after rapid degradation of that piece of DNA. So the piece of DNA which is making the laureate, this is finally degraded.